Hi everyone, welcome to my channel where you can access videos on topics from biology and biochemistry. If you're enjoying these, please hit the like button and share it with your friends. Now, as you all know, carbohydrates are an important biomolecule that you find abundantly in nature. And isomerism is one of their very key properties. So in this video, we'll look at the different types of isomers and isomerism that you find in carbohydrates. And we'll also see how it's contributing to the function of carbohydrates. We'll start with knowing the meaning of isomerism and the difference between structural and stereoisomers. Carbohydrates can exhibit different isomeric forms because of their chiral carbon positions, which includes isomers like enantiomers, diastereomers, anomers, and epimers. We'll study in detail about each one of these. Now, isomers are molecules that have the same chemical formula but differ in the organization of atoms within the molecule. This can lead to them having different physical or even chemical properties. For example, as you can see here, I've shown you three isomers of pentane. Now, all three of them have the same number of carbon and hydrogen atoms. However, as you can very clearly see from the diagram, they are arranged very differently. And because of their different arrangements, they have different melting points and boiling points. Each isomer of pentane, in fact, has a distinct role and an application in the industry. And it's all because of the way they are arranged. In case of carbohydrates, carbohydrates are characterized by a chiral carbon. A chiral carbon is a carbon that is attached to four different atoms or four different group of atoms. So the configuration that's called as chiral is what allows it to lose all the symmetry, and these chiral carbons are also called as stereogenic carbons or asymmetrical carbons, like you can see over here in the third picture in the D galactose. As you can see, these carbons, say you can look at this carbon over here. This carbon is attached to four different groups. So when all the four groups that are attached to a carbon are different, such a carbon is called as a chiral carbon, and such an asymmetrical carbon is what allows the carbohydrates to have multiple types of isomers. So we'll be looking at each of these isomers. Let's first start with structural isomers. Structural isomers, example glucose and fructose, a classical example, are atoms that are bonded differently. So the arrangement of atoms is completely different. As you can see here, glucose has an aldehyde group as seen over here, a CHO group, whereas fructose has a ketone group, that's a C double bond O group. But if you count the number of carbon atoms, count the number of oxygens in them, count the number of hydrogens in them, it's exactly the same. Both are monosaccharides and both are very easily absorbed by the body, but they have different structural formulas. Identical molecular formula, but different structural formula. They are arranged very differently and their functional group is different, which makes them structural isomers. So glucose having an aldehyde group is called as an aldose sugar, whereas fructose having a ketone group is called as a ketose sugar. We'll talk about aldose and ketose sugars more in our other videos. So these are an example, or glucose and fructose are a very good example of structural isomers, which are isomers where the atoms are bonded differently. The arrangement of atoms is completely different. Let's now look at a very confusing yet very interesting type of isomers that you find in carbohydrates. Those are called as the stereoisomers. Now, stereoisomers are those where the arrangement is same, where the order of atoms is exactly the same in both the molecules. However, when you look at the 3D structure, their 3D organization is different around at least one asymmetrical carbon atom. As you can see here, this is a chiral carbon example that I've given you. So this carbon is now bound to four different groups. You have on one side there's a CHO group, that's the aldehyde group. On one side it has OH, that's the alcohol group. On one side it is having CH2OH on, and the last one is attached to a hydrogen. So these isomers, these stereoisomers are differing in their spatial arrangement, in the 3D arrangement around that chiral carbon. And that is why they are forming different types of stereoisomers. So what we're going to look at in the next few slides are all different types of stereoisomers. That is isomers that are differing, that have the same structural formula, but are differing in the spatial arrangement. Now, these stereoisomers are described in carbohydrates using the DL notation. Apart from this, you would have also heard of RS notations, or you would have heard of plus minus notations, or even DL notations. But when we come to carbohydrates, we usually talk about notations, or the notations which are most commonly used are the capital D and capital L notations. Now, these D and L notations in the carbohydrates are just referring to the direction of the hydroxyl group or the alcohol group or the OH group. So, it's the direction of the hydroxyl group in the chiral carbon that's furthest away from the aldehyde or the ketone group. So, you first look at the molecule. In the molecule, look at where the functional group is or where the aldehyde and the ketone group is. Like you can see over here in L-glucose, we have the functional group over here. You look at the carbon that's furthest away from it. So you're looking at the at this carbon, and at that carbon, you check that's the last chiral carbon. So you look at the last chiral carbon and you see which side the OH group is facing or the hydroxyl group is facing. If the OH group is facing to the right side, then we call that as a D form of that particular sugar. And if it is facing to the left side, we call that as the L form of that sugar. So in this case, what I've shown you is the D and the L forms of glucose. All the other atoms, all the other groups stay exactly the same. It is only the hydroxyl group which is facing either to the right or the left side. And this is in the last chiral carbon away, furthest away from the functional group. In this case, the functional group is aldehyde. If I'm showing you a keto sugar or if I'm showing you ketose, then the carbon, that's the chiral carbon that's furthest away from the keto group is what we'll be looking at. So this is what D and L notation stands for. Now the rules for RS notation or plus minus or DL, the small d and L, all the rules are different. So you could have a, we could have a D compound, for example, even in amino acids we'll be seeing this. We could have a D compound that can be either an R or S type of compound. So for open chain common sugars, usually the D-glucose configurations are usually R, and the L-glucose or L-chain sugars are usually S configuration. But again, this is not a blanket rule. It can change. The one thing that we do need to keep in mind is that all the biologically relevant carbohydrates are of D configuration. This is what we have found out. So almost all the carbohydrates that we'll be coming up with or we'll be encountering are all of the D configuration or D notation. So how many stereoisomers are possible for a carbohydrate? Many. The reason is because around every chiral carbon, each chiral carbon, you will have two different stereoisomers that are possible. So for every carbon that's chiral in a particular molecule, I could have two possible stereoisomers because there's no plane of symmetry in around that. It is a chiral carbon. There are four different groups that are attached to it. So in a carbohydrate which has multiple chiral carbons or multiple stereo centers, I would definitely have very many number of stereoisomers. For example, in glucose, there are 4 chiral carbons, which means if I need to find out how many stereoisomers are there, I just need to apply the formula 2 raised to the power 4. That would be equal to 16 stereoisomers just for a molecule which has 6 carbons. This is how you would know how many stereoisomers are present. But what are these stereoisomers, or what are the names, or how do they exactly behave? So there are two major categories of stereoisomers. We have enantiomers and diastereomers. And then diastereomers are further divided into epimers and anomers. So we'll be looking at each one of these in detail. Remember that enantiomers are also stereoisomers. It's just that they differ in configuration at every single chiral center. We'll look what enantiomers are all about in the next slide. Enantiomers are stereoisomers that are non-superimposable mirror images of each other. What does this mean? Let's break it down. Mirror images, because if you see each and every group around every chiral carbon in this molecule has been flipped. As you can see over here between D and L glucose, you can see that at every single carbon, the groups have been flipped. Like if you have H on the left side and OH on the right side, when you see L-glucose, it's exactly the opposite. At every single chiral carbon, that's the point to take home over here. So at every single chiral carbon, when you flip the compounds or when you flip the groups, that results in a mirror image. But then if you keep the mirror image one on top of the other, like how you keep your two hands one on top of the other, you will see that it's non-superimposable. You can't superimpose it. It doesn't look the same. They are exactly opposite of each other. Now this leads to them having identical physical properties like melting point or boiling point or solubility. However, though they have identical physical properties, they interact differently with plane polarized light and with other chiral molecules. So the property of enantiomers is that they are optically active. That's one property of enantiomers that you need to keep in mind. Now, if you see the D and L isomers as given over here, they are different not just in the last chiral carbon, which is what we spoke about a little earlier, but they are different in every chiral carbon. That is why they are forming a set of stereoisomers. They are forming a set of enantiomers. This means that when we say that they have, they are optically active, but they are having different optical activity, it means that they can rotate plane polarized light to a certain degree. However, they are rotating it at different angles. So if one enantiomer is rotating plane polarized light at an angle of, say, plus 34 degrees, the plus is just to show that it is rotating it towards the right or dextro-rotatory, then its enantiomer would rotate the plane polarized light at the same angle of 34 but negative 34. Negative here indicates that it is rotating the light towards the left or it is levorotatory. So they have the same magnitude of rotation, both the enantiomers. It's just that they are rotating it in different directions. So that is one of the most important things to know about enantiomers, that they are optically active. However, they are rotating the light in opposite directions and they are non-superimposable mirror images. Let's now look at the other type of stereoisomers. Anything that's not an enantiomer is called as a diastereomer, which means to say there has to be at least one chiral carbon or one chiral center which is opposite, but not all, because if all the chiral centers are opposite, we call them as enantiomers. So there has to be at least one. It could be two, it could be three, it could be four, but not all. In that case, we call such stereoisomers as diastereomers. So these are definitely not mirror images of each other, because not all the chiral carbons are inverted or not all the chiral carbons are arranged oppositely. Only one or two, maybe one or more than one, are arranged differently. Hence, they are definitely not mirror images of each other. For example, as you can see here, the aldopentoses each contain three chiral centers or three chiral carbons. This leads to D-ribose having three different diastereomers. What are the three different diastereomers we have? We have D-arabinose, D-xylose and D-lysose. All three of them are diastereomers of D-ribose. How is that happening? Let's have a look. So if we look at the carbons, they have been numbered from 1. We always start with the functional group. And then as we go down, we have labeled them or numbered them from 1 to 5. If you look at arabinose, in between arabinose and ribose, it's the second carbon where the chiral center is different. So at the second carbon, the groups are inverted. The groups are arranged differently. So we can say that arabinose is the diastereomer of ribose at C2 or at carbon 2. It goes something similarly for xylose and lysose as well, along with ribose. So they are all diastereomers of each other. Now, there are two particular types of diastereomers that we'll talk about. Those are epimers and anomers. When two diastereomers are differing from each other at just one stereocenter, those are called as epimers. So, in a molecule, say for example, if there are 6 carbon atoms and only one of the chiral centers is differing from, in both the molecules or in both the isomers, such a type of diastereomer is called as an epimer. So, the point for you to remember here is that all the epimers are examples of diastereomers, or all the epimers are actually subtypes of diastereomers. Now, the example I've shown you over here of epimers are the D-glucose, D-galactose and D-mannose. Very, very common examples. Or in the earlier example that we saw, ribose and arabinose, they are also examples of epimers. Why is that? Because they are differing only at one carbon atom. Now, for example, if you see over here, in between the D-glucose and D-mannose, both are aldohexoses, both have the same molecular formula, but if we look at their structure, we'll see that the position of the hydroxyl group is varying. At which carbon? At C2, at second carbon. So we always start numbering the molecule first, starting at the functional group, which is the aldehyde group over here, and then this is the second carbon. So if you look at the second carbon between mannose and glucose, the H and OH, the hydroxyl group and the hydrogen group, has been interchanged at the second carbon. So we can say that mannose and glucose are epimers of each other, or they are C2 epimers of each other. Why C2? Because they are differing at the second carbon. Or in other words, glucose and galactose are C4 epimers of each other. Why is that? Because they are differing only and only at the C4 carbon. So one thing for you to remember is that epimers are differing at just one chiral center in the entire molecule. That's why they are subtypes of diastereomers. Now, a special case of epimers which are differing in the position at the anomeric carbon are called as anomers. So, anomeric carbon is the carbon that's involved in the cyclic formation, or in the formation of the cyclic group or the cyclic ring-like structure. So anomers can actually now be considered as a subtype of epimer, and they're commonly associated where you have cyclic sugars, or where the linear form of sugars that we saw earlier undergoes a cyclization. So you have a linear form of a sugar that undergoes a cyclization. It can form different types of structures, which we'll look at a bit shortly. But then once it forms the cyclic structure, that cyclic structure is where you see the anomer, or that's where you can identify the anomers. Now, when the sugar molecule undergoes this intramolecular cyclization at the C1 carbon, like you can see in the diagram given over here, at the C1 carbon, or in cases of when you have keto sugars, it's usually at the C2 carbon. So either C1 or the C2 carbon, that is where the arrangement or the configuration differs, and such isomers, such stereoisomers are called as anomers. So the anomeric carbon is the carbon where the aldehyde, or which is bearing the aldehyde or the ketone functional group in the open chain form. That is the one which is cyclizing, or that is the one which is forming the cyclic form. That is why it's called as the anomeric carbon. So what are these cyclic forms? Now monosaccharides, which have 5 or 6 carbon atoms, when they are in an aqueous solution, they form cyclic structures via an internal hemiacetal or hemiketal formation. So it's happening within the same molecule. Now I'll tell you what hemiacetal or hemiketal is normally, but here it is happening within the same molecule. Now normally hemiacetal is formed when an aldehyde reacts with an alcohol. So let me show you a reaction. This is an aldehyde, RCHO. This is the aldehyde. It's reacting with an alcohol. So we have ROH. And when this reaction occurs, you have a hemiacetal being formed. That would be RC. We would have OR over here. This is OH over here, and the hydrogen group. Or how would a hemiketal be formed? Hemiketal is formed when you have ketones that are reacting with the alcohol. So we would have a ketone like this, RC double bond OR, reacting with an alcohol like we saw in the previous one. And the reaction formed over here, or what is formed at the end, is what we call as a hemiketal, which would be like this. So we have R, C, there's OH over here, there's OR over here, and we have R group at the bottom. So this is what we call as a hemiketal, and the previous one, what I had shown you, is called as a hemiacetal. Basically, when an aldehyde reacts with an alcohol, or when a ketone group or you have a ketone reacting with an alcohol, you get these structures. But here we don't need an alcohol from outside. The carbohydrate itself is having OH group, or it is having the hydroxyl group. So we have carbons like, for example, the C1 aldehyde. The C1, the carbon 1 aldehyde is forming, it is reacting with the C5 hydroxyl group to form an intermolecular or intramolecular hemiacetal formation. Or in the ketones, it is the C2. So for ketone sugars, it is the C2 carbon which is undergoing this reaction. In case of the aldohexoses, example glucose, it is the C1 carbon that is undergoing this formation. So these are reacting with the C1, or the C2 carbons are reacting with the C5 hydroxyl group to form the structure that we have seen now. Now if it is a glucose, or if it is an aldohexose, it would form always a pyranose kind of a sugar which is resembling a pyran ring. But if it is a keto sugar, it can form, like for example fructose, it can form a 6-membered cyclic hemiacetal, that's called as the pyranose. Or fructose can also form the structure that's given over here, which is 5-membered, that's called as a furanose sugar. So the 5-membered cyclic structure is called as furanose because it is resembling a furan ring. Whereas a six-membered structure, cyclic structure, is called as a pyranose because it resembles a pyran ring. So aldohexoses always would form a pyranose kind of formation, whereas the ketohexoses like fructose can either give you a six-membered cyclic structure, that's a pyranose, or it could give you a furanose. The more common one that you would see in case of fructose or in case of ketohexoses is usually furan rings. So furan rings are formed, they are the ones that are predominant when you see them in most fructose derivatives. But even pyran rings are formed in case of fructose, which are seen free in the solution. So this is what anomer is all about. Anomer is an example or is a subtype of epimer. To sum it up, these are the different isomeric forms of carbohydrates that we have seen now. We have seen what are the structural isomers. We have seen what are the structural isomers are also called as the constitutional isomers. We have seen the stereoisomers, which are just differing in the 3D arrangement or the spatial arrangement. Under that, we have enantiomers and diastereomers. And diastereomers are again divided into two subtypes. Those are epimers and anomers. Let's do one question based on this. So we can see that the yellow box here contains the structure of a pyranose aldohexose. Pyranose because it's a six-membered ring. And beneath that, I've given you four different structures of stereoisomers, which are labeled A, B, C, and D. Now, the questions which are given below are based on this structural relationship between the main molecule and the four isomers that are given below. You can pause the video over here, take a moment to solve the answers for this, to solve out each of these questions, and I shall tell you the answers in a few seconds. For the first one, which of the formula from A to D is an anomer of the reference structure? You can see very clearly that C is the only anomer for the reference structure that's given in the yellow box. Whereas the second question is which of the formula are, from A to D is an epimer of the reference structure. There are two epimers given over here. Both A and C are actually epimers of this molecule. And the last one, which of the formulae is a diastereomer of the reference structure? All four of them are diastereomers. None of them are enantiomers. All four of them are examples of diastereomers of the reference structure. If you've not understood how I have arrived at the answers for any of these three, you can always revisit the previous parts of my video, or you can ask for the expanded solution in the comments and I'll definitely help you out. With this, we have come to the end of today's lecture. We need to know that isomers of carbohydrates are really very important because, two isomers can have the same chemical formula, but they have different chemical structure, which contributes to their properties. So they could have different properties in the molecule. And carbohydrate enantiomers are, often precursors of many compounds that you find in the cells. So it could be in the plant cell, in the microorganisms, insects, animal cells. All of them are utilizing both types of carbohydrate enantiomers, but we know that the major biomass or the excrements that are coming from these cells are dominated by the D enantiomers. So the knowledge of isomers is very, very important for us to know how they are playing a role in the cell and how they are contributing to the cellular structure. I hope this video was useful to all of you and see you in the next one as well. Thank you.